Hello my dear Slate champions, I welcome you all to score 99% in 99 days. Today is day 62 and you already know the importance of this particular day because we are going to start solving sample papers. So I hope the concepts are done, theory is done, everything is done, and now it's time for practicing questions, which is very important. So if you are someone who is wanting to revise the chapters which you have already read, I'll suggest you to revise by solving question instead of going through your notes, because that is kind of boring. Sometimes when you go through your notes you feel sleepy and things like that. So I don't want you to do that. You can do that later on when you have got very less time, okay, towards the time when you are very near your exam. As of now, I will recommend you to practice more and more question. And here is sample paper 1, my dear children, and it was published in the CBSE portal for this year, that is 2024-25 batch. So I don't want you to miss it, and here we will be solving questions exclusively from the biology part. Okay, so let's get started, and I hope you know that biology has the most amount of weightage, probably because it has high number of chapters as well. Okay, so you will be solving 9 questions which are 1 marker questions. Okay, 3 of them 2 marker, 2 of them 3 marker, 1 of them 5 marker, and 1 of them 4 marker. In a total, it's 30 marks. So out of 80, 30 marks are coming from biology itself. Rest 50 marks. Rest 25 or probably 27 something. Okay, would be from chemistry and physics. So let's get started without wasting any bit of your time. Section A, my dear children, consists of 1 marker question. So you would be having MCQs and you would be having 4 assertion and reason. In the case of biology, you will get 2 assertion and reason questions. Okay. Chalo. Question number 8 directly. Rest of them are from chemistry, I believe so. Yeah. So during cellular oxidation of glucose, okay, that is respiration, ATP is produced along with formulation or basically formation of other products in this reaction. Which of the following events is associated with production of maximum ATP molecules per molecule of glucose during the process? Synthesis of ethanol, lactic acid, carbon dioxide. So if you know, you know. What happens, my dear children? The glucose is broken down to something that is known as pyruvate. Now the pyruvate is given an option. In the cell, enough amount of oxygen is there. The pyruvate enters the mitochondria, undergoes a cycle of conversion and things like that. It undergoes Krebs cycle, okay? And at the end, you get CO2 plus H2O plus ATP. Okay, this is what we were aiming for. Now, suppose this is when oxygen is there. Suppose oxygen is not there. At this point of time, in our body, the pyruvate basically converts into something else that is known as lactic acid, my dear children, and also again ATP. Fine. Again, this is when oxygen is not there. Okay. Again, when oxygen is not there, this happens in the case of yeast, where what is happening? Ethanol is being produced, and then CO2 is also being produced, and then ATP is also being produced. So, ma'am, 3 of the places ATP is being produced. The thing is, during this type of respiration, that is known as aerobic respiration, where oxygen is there, my dear children, there are so much ATP molecules which are being produced, almost 38 of them. Whereas when you talk about this case, there will be 2 ATP molecules which will be produced. So 38, 2, it's a huge lot of difference, right? So in the synthesis of what do you see more amount of ATP molecules? Ethanol and yeast? No, dear children, it will be really less. So this is wrong. Lactic acid in the muscle cells. In the muscle cells, when anaerobic condition is there, lactic acid is being produced, which causes muscle cramps. Yeah, and also ATP is being produced. So this is also wrong because less amount of ATP is being produced. Our body thinks we have to anyway produce ATP. It needs, energy is needed, right? But what happens is that it is also producing lactic acid, which is going to cause the muscular cramps. Now carbon dioxide in yeast cells, again, that is something like this. ATP is not much produced, but carbon dioxide in the case of human cells, my dear children, when this is produced, that means along with it, lots and lots of ATP is also going to get produced. Fine, and this is known as aerobic respiration. What do you call it? Aerobic respiration. So your answer becomes D. I hope you got the 1 mark. Now, Next question. During which of the following stages of the circulation of blood in a normal human being, the oxygenated blood is pumped to all the parts of the body? During which stage? So, I hope you know we have 4 boxes. Okay, this is the box. Left atrium, left ventricle, right atrium, right ventricle. Okay, you get oxygenated blood from the lungs and it goes to the left atrium. Left atrium says, "Lao, bring in oxygen." So, oxygenated blood comes over here, oxygenated blood. This blood goes to left ventricle. From left ventricle, it will be going to the body parts because the body parts are crying for oxygen. Remember, oxygen is needed for much formation of ATP. That is why it is going this side. From the body parts, as a return gift, body part will be giving deoxygenated blood, my dear children. Deoxygenated blood is given again to the atrium first, then to the ventricle, then from ventricle it goes to the lungs for oxygenation, and the whole cycle repeats again and again. So, the oxygenated blood is pumped to all parts of the body when contraction of left atrium happens. When contraction of left atrium happens, it is going to send to the left ventricle. Contraction of left ventricle happens. When contraction of left ventricle happens, it is going to send to the body parts. So, this is correct. But let's see the other option. Relaxation of the right atrium. Relaxation as in when they are totally normal. Do you think at that point? No, at that point deoxygenated blood is going to fill. That is why it is relaxed. Now, relaxation of right ventricle, again, no, that is not the answer. So, what is your answer, my dear children? Your answer is B. Okay. Children, my hair is not being straight today. Yeah, cool. Next question. Which of the following adaptations in herbivores helps in digestion of cellulose? So, I hope you know that. In the case of herbivores, they are feeding on plant products, and the plant cells which are there, they have thick cell wall, my dear children, and this cell wall which is there, it is made up of something that is known as cellulose. And for breaking down cellulose, it takes a huge lot of time. So, if you want to break it down properly, you should have time to spend it. Okay, you should have enough time to spend on it, right? So, this process is basically done in the small intestine of the herbivores, and they have really long small intestines such that maximum breakdown of cellulose can be taking place. Okay, so I'll say longer large intestine, no. In large intestine, there is no breakdown of big things. There is only absorption of water. Okay, smaller large intestine, no. Smaller small intestine, that means there is only very less time to break down things, right? No. Longer small intestine, this becomes the correct answer. If it is longer, you have lots and lots of time for breakdown of cellulose. This is the correct answer. Okay. Now, next question. There was a cerebellar or basically cerebellar dysfunction in a patient. Which of the following activities will get disturbed in this patient as a result of this? If the cerebellar stands for what? Cerebellum, my dear children. If the cerebellum which is there, which is the part of your hind brain, my dear children, if it is not working properly, what will be getting disturbed? So, I always make you remember like this. LL, it looks like a straight thing. So, basically it is for cerebellum, it is mainly responsible for your posture and balance. And people who drink a lot of alcohol, this alcohol actually affects the cerebellum, which is why they are not able to maintain a proper balance. You can remember like that. So, cerebellum is responsible for your posture and balance. Cool? This becomes your answer. What about salivation? Salivation is an involuntary action. It is not under your control. It is controlled by, okay, whenever involuntary things are there, it is controlled by medulla oblongata, which is also a part of hindbrain. Now, when you talk about hunger control, hunger is controlled by hypothalamus. Hunger, all your physiological needs are controlled. Hunger, sexual drive, thirst, everything is controlled by hypothalamus, which is there in your forebrain. Okay, regulation of blood pressure, again by medulla oblongata and stuff like that. Works? So, this is the answer. Fine. Going ahead, what do we have? Okay, in snails, individuals can begin life as male, and depending on environmental condition, they can become female as they grow. This is because, so you think an organism is able to change its sexual orientation. How can it do that? How can it not sexual orientation? Basically the sex. How can I do that? Just give me a second. I'm getting a call. Quick little. Okay. So this is because male snails have dominant genetic makeup. Female snails have dominant genetic makeup. Expression of sex chromosome can change in a snail's lifetime. Sex is not genetically determined in snails. This can be correct thing, right? If it is genetically determined, my dear children, do you think it can change? No, it cannot change. So this becomes the answer that why snails can change their sexes. So sex is not genetically determined in their case. Okay? So over here, this becomes the answer. Identify the incorrect statement. The energy available to the producers is maximum. You have to find what the incorrect statement. So underline incorrect, because sometimes you go try to find the correct one and everything might seem correct to you. You get it? It's because everything is correct. Only one thing is incorrect. So you have to find the incorrect statement, underline that. So it is said that energy that is available to the producers, it is really high. Why do you think so? First, think about that. So it's because the sunlight which is there, the plants which are there, the producers, why they are known as producers? Because they are the one who produce the food, my dear children. They are the one who make organic content. How? By using the light energy. They do so by using the light energy. So they're the first organism to get the light energy directly. How much amount of light energy? 1% of the entire light energy, they are going to fix it. Okay, 1% of the entire light energy, they are going to fix it. And now they have little amount of energy. They are going to pass it to the next organism, but only 10% they will pass. Rest 90% is lost. Some of it will utilize, some of it is lost. Which is why the producers will be having a lot of energy in them, because they took it directly from the sun. How much? 1%. And also they are basically they are also giving energy, but not too much, only 10%. Get it? That is the reason. Now we have to find something that is not compatible with this. So it is first trophic level which absorbs 1% of light energy directly from the source. This is correct. It utilizes most of the chemical energy for its own respiration, growth, reproduction, movement, etc. That is also correct. It utilizes 10% of the light energy and transfers the rest to the next trophic level. Does it utilize 10%, my dear children? No, it is 1 to 2%, that's all. It transfers only 10% of light energy to the next trophic level. That is also correct. So this becomes the wrong answer. It is not that it takes in 10% of the light energy and transfers the rest, whatever rest is there, to the next organism. It is not like that. Okay, it takes 1 to 2%, it gives 10%. Fine. So this becomes the wrong statement. This is the incorrect one. So let's give it C. Now, next type. Which of the following is not a role of decomposers in the ecosystem? Again, not a role. Underline that. What does decomposers not do? So decomposers are responsible for breakdown of large things into smaller things, such that the important nutrients which is there can be taken up by organisms such as plants and stuff like that. Okay, they are going to make the soil quality really well. They are going to break down the dead and decaying thing. They are going to break down the waste substances, everything. Okay, that is their job. We have to find what is mentioned over here and what is not their job. So natural replenishment of the soil, this is their role. Enrichment of oxygen in the atmosphere, they have nothing to do with that. They are not going to enrich the amount of oxygen. How can they do that? So that is wrong. Okay. Waste decomposition, yes, they do that. Breakdown of dead remains, they also do that. But this is not something that they do, my dear children. This is not their role. So which is why I'll say B is the correct answer for this thing. Cool. Now, next question. Which of the following is, okay, assertion and reason question, okay. Assertion says, generally, the number of chromosomes in a cell and in a germ cell is not the same in species. So, number of chromosome in a normal cell is 46. Number of chromosome, this is a normal cell. Number of chromosome in a germ cell, that means you're talking about gametes. In them, it is not the same. Yes, it is not the same. I truly agree with that. Why so? When the two germ cells combine, they restore the normal number of chromosomes in a species. This is the reason. Yeah, this is the reason. That is, the germ cell, if one has 23, it has to fuse with the other germ cell which has 23. Together when they come, they are going to make a cell which has 46 number of chromosomes, just like a normal cell. So this is their rule. This is why they are halving the number of chromosomes. Suppose it was just having 46 from 46, 46 plus 46, 92. Then it would have been a problem. It would deviate from the normal nature of our cells, right? It will make double the amount of proteins and everything. We do not want that, which is why halving is really important, because anyway these gametes are going to come together and are going to make something like this. So generally the number of chromosomes in a cell and in a germ cell is not the same in a species is correct. Reason is that when two germ cells combine, they restore the normal number of chromosomes in a species. That is also correct. So both A and R are true, and R is the correct explanation of A. Okay? Cool? I hope you are understanding that. Now, this is the correct answer. Assertion says if the lions are removed from a food chain, it will not affect the food chain. Suppose in a food chain you have plants. Plants are eaten by deers and deers are eaten by lion, and I am removing lion. Okay, I am going to say that even if I remove the lion, it is not going to affect the food chain. However, if the plants are removed from a food chain, it will disturb the ecosystem. Just because the plants are making the food and lions are not, does not mean that the lions are not important. These lions which are there, suppose these lions which are there, if they are not there, the deer will be increasing. The deer population will increase. If the deer population increases, it will be eating on more and more plants. The plants will die. Will you have producers, my dear children? Do you think ecosystem will maintain its balance? No. So even a predator is really important. Otherwise, the prey species which is there, it is going to increase in number and it is going to cause a huge lot of trouble anyways. So you cannot say that lion is not important just because they are not making the food. So this is a wrong statement. Okay, let's see the reason. Plants are producers who can make food using sunlight, while lions are consumers. The statement is correct. The statement is absolutely correct, but it has nothing to do with the assertion. So what will I say? Assertion is false, and this particular reason is what? True. Assertion is false, and the reason that I see over here is true. This is the correct statement. This is not the correct reason for the assertion, but this is the correct statement. I hope you get it. Okay? Cool. Now, going ahead with the next question, that is section B. It's a 2-marker question, my dear children. Let's get started. So over here, what do we have? Differentiate between alveoli and nephron on the basis of the following points. What do you mean by alveoli? Alveoli is there in the lungs. Okay, you might be knowing you have bronchi. Bronchi breaks down into bronchioles, primary, secondary, tertiary, and everything like that. And at the end, what do you have, my dear children? You have alveoli. This alveoli, they are richly supplied by blood vessels. They are richly supplied by blood vessels. What do they do? Basically, the blood carries carbon dioxide. Okay, it takes in from different parts of the body. It is carrying carbon dioxide, bringing it up over here, and it gives the carbon dioxide to the alveoli. Alveoli is somewhere where when we breathe in, we take all the oxygenated thing and everything inside, and it is now here inside the alveoli. Okay, so alveoli has oxygen, blood has carbon dioxide, oxygen goes to the blood, carbon dioxide goes here because of the pressure difference that is there, right? That is how exchange of gases take place. Now the structure and the location, you know, alveoli is a balloon-like structure, and it is present in the lungs. What about nephron? Okay, nephron has a cup-like structure, then a tube-like structure and everything. Where is nephron present, my dear children? It is the basic functional unit of your kidney. So this is the location. Now what about the function? The function of alveoli is to basically exchange the gases. That is oxygen and carbon dioxide. What about a nephron? Nephron's function is to remove what? Nitrogenous waste and selectively reabsorb. What do you mean by reabsorb? As to take it back. Okay, selectively as in you are selecting and taking things back. Selectively reabsorb the important things. For example, you want water, you want ions, you take them back. No problem. Okay, it is not going to be excreted out. If your body is not needing it, then you will probably remove it. So removal of nitrogenous waste or any type of another unwanted stuff is done by nephron. Cool. How do you frame the answer? You can frame like this. Structure and location, balloon-like structure present at the terminal ends of the bronchioles in the lungs. They have explained it really well. And children, this is not like I made the answers. It is given by the CBSE itself in their marking scheme. So you should be knowing it really well. What about nephron? Nephron is a tubular structure present in the kidneys. Now, what about the function? Exchange of gases. Over there, nephron, filtration of blood to form the urine. This is a direct thing. Okay, it helps in what? Filtration of blood. It is helping in filtration of the blood by removing the nitrogenous waste and selectively absorbing the important things. So directly you write the answer and you can explain a bit also. Okay, filtration of blood is really important. This particular term that is given over here, it is highly important. Cool? Works? Chalo. Next, attempt either option A or B. List the steps for the synthesis of glucose by the plants. Okay, list the steps for the synthesis of glucose. That means, what is glucose, my dear children? Ma'am, I have not studied this. How did it come? Glucose is basically a carbohydrate. So you are asked to explain the synthesis of carbohydrate. That means you are asked to explain the steps that are involved in the process of what? Photosynthesis, my dear children. Okay, what special feature is found in desert plants related to this particular process? So what are the steps of glucose synthesis? First step is what? Absorption of sunlight by the chlorophyll pigments. First thing, the sunlight has to be absorbed by the chlorophyll pigments, right? Once they are absorbing the sunlight by the chlorophyll pigments, you are going to convert the light energy into what? Chemical energy, that is in the form of ATP and things like that. And while you are doing so, you are also breaking down the H2O, the water that you have taken inside, to oxygen as well as H molecules, that is hydrogen molecules. This oxygen is something that you release during photosynthesis process. Okay, in photosynthesis, what do? You do carbon dioxide, H2O comes together to form C6H12O6 plus O2 plus H2O plus ATP. This O2, where does it come from? From the breakdown of water. So first is absorption of sunlight by chlorophyll. Second is conversion of light energy to chemical energy, and also breakdown of water. This is known as photolysis of water. Okay, while you do so, there are so many electrons being released, and these electrons are going to help in the formation of ATP formation. That is the thing. Now there is a third step, reduction of carbon dioxide to carbohydrate. Reduction of carbon dioxide to carbohydrate. Now how it is different in the case of desert plants, my dear children? Desert plants, you know, they are in a very dreaded condition. Okay, there is no enough amount of water in them. So in them you cannot just open the stomata and keep it like that. If you do so, what will happen? The water will be lost. So in them, daytime, in the daytime they do not have an option of opening the stomata. At the night time, what do? Is they open the stomata, take in the carbon dioxide and keep it safe. Okay, anyways, you need the carbon dioxide for the production of carbohydrate. So what do? Basically, you take in the carbon dioxide and keep it safe. You convert it into different molecule which you call as malate, and you store it inside the vacuole. Now in the daytime, in the night time this is happening. If I said daytime, please forgive me. In the night time this is happening. They are opening, taking in and keeping it safe. How? They are converting the CO2 to malate. Now in the daytime, my dear children, this malate is going to break down again into CO2, and the CO2 is going to reduce into this thing. But how, ma'am? Why in the daytime this can't it happen in night time only? Children, the chemical energy that is made up over here, it's needed up over here. The chemical energy which is made during the daytime, that is when light energy is converted to chemical energy, that particular process basically is needed up over here. Got it? So that is why this is different. And that's all. This is how the answer looks. So just have a quick read of it. Okay? Okay, now it's time for the next question, that is, explain the role of the following enzymes in the process of digestion of food in humans. So these are a set of enzymes, you have to tell its role. Starting from salivary homeostasis, my dear children, remember, it's the first enzyme which is going to break down the food. And where is this particular enzyme being produced? It is being produced in our mouth. In our mouth. Okay, and it is going to break down a complex carbohydrate which is starch into what? A simpler form of carbohydrate which is what? Maltose. Cool. So with this you get one more information, that the digestion process starts to take place in our mouth. We think that mouth is just for chewing, all the mechanical action and stuff like that. There's no enzyme that will be working. But children, please remember there's an enzyme, and it's a very important enzyme, that is salivary amylase, the first enzyme that is going to act on starch, which is what? A carbohydrate. So breakdown of carbohydrate starts in the mouth, or breakdown of food starts in the mouth itself. Talking about pepsin, P for pepsin and P for proteins. Pepsin, which is a hormone being produced in our stomach, is responsible for the breakdown of proteins into peptides, my dear children. Protein into peptide. Now you only think, okay, suppose we had some food, okay, it went inside. Now there might be carbohydrate, there might be protein, there could be fat, a lot of things. So for different stuff, you have different enzymes as well. Okay, so pepsin is an enzyme that would be specifically acting on what? Proteins, and break it down into peptides. But pepsin is not being active all the time, or basically there is this particular enzyme that you see is not being produced as such. Instead, there is another enzyme which is being produced, which is known as pepsinogen. Pepsinogen is an inactive version of this particular enzyme. Only when there is perfect pH, that is acidic pH, that's when the pepsinogen is going to convert into pepsin, and now this pepsin is going to act on proteins and break it down into peptides. So that is the role of pepsin. Now again you have trypsin. Pepsin, trypsin, all of them, they are again going to act on proteins only, and they are going to break them down into what? Peptides. The only difference that they have much more differences, but yeah, according to your NCERT, the only difference that they have is trypsin is basically secreted by a gland, and that gland's name is, it is a heterocrine gland. Okay, it's an organ only, but you call it as an heterocrine gland because it has two functions. It is going to release enzymes, it is going to release hormones. Can you guess the name? Can any one of you guess the name? What is that particular organ that is going to act like an exocrine gland that is releasing enzymes, and also an endocrine gland that releases hormones? And the famous hormone is insulin, my dear children. It is none other than pancreas. So your pancreas are responsible for the production of trypsin. Again, this will be in the form of trypsinogen, which will be inactive. It is later formed active, but this time in under alkaline pH. This needs acidic pH, pepsin needs acidic pH, trypsin needs alkaline pH. Now you have lipase. The name suggests lipase is for lipids, my dear children. It is for lipids. It is going to break down. Fat is a type of lipid, right? It is going to break down fat into simpler substances like fatty acids and glycerol. So fat is made up of fatty acids and glycerol, my dear children. These fatty acids can be in a combined state, which will be broken down again in the small intestine. Remember, the final digestion, the final breakdown of all these things that you see over here, they will also have their simpler forms. It will be happening in the small intestine. So lipase basically acts on lipids, and fat is such thing. So it will be broken down to fatty acid and glycerol. Now let's just see the answer, how it is given in the marking scheme. Okay, salivary amylase breaks down starch, which is a complex molecule, to sugar. What sugar, my dear children? It is maltose. Okay, now pepsin helps to digest proteins in the stomach into what? Peptides. Trypsin again helps in digesting proteins to what? Amino acid. Okay, and then that is basically peptides, and then peptides are broken down to amino acid. Well, amino acids are not being made up here, it's made up in the small intestine, but okay. Now lipase breaks down of emulsified fat to fatty acid and glycerol, my dear children. Okay, it is going to break down fats. What do you mean by emulsified fats? Emulsified fats as in, there's a big fat globule, imagine. If you break down the big fat globule into smaller fat globules, you call this as emulsified fat. And who is responsible for breaking down this big fat globule? It is bile juice, and bile juice is made in the liver, my dear children. Remember that also. So lipase cannot act on big things. So to make their work easy, bile juice, which is secreted by the liver, is going to break them down into smaller things, fat globules, and lipase is then going to act. Okay, which is why it is written, it is acting on emulsified fat to produce what? Fatty acid and glycerol, my dear children. With this, we are completing this question as well, which is of 2 marks. Now, let's get ahead into the next question that we are having. The cartoon below addresses a growing concern. So what is happening over here? Okay, these are some plants that is mentioned over here. I think it's cabbage, yeah, and this guy is putting something onto it. What is that something? Let me zoom it and show it to you. Over here you see pesticide. He is spraying pesticide so that pests do not come and act. Okay, and then later it is shown that basically whatever substance is there, it is going to seep. It is near a river or basically water body, and everything which is there is getting seeped inside what? The aquatic water body that you are seeing over here. Now, why is it a concern? What impact will the process shown in the image have on humans if they occupy the last trophic level, and explain the reason for it. So, what do you think is a problem? Over here, please remember one thing. So, when you put pesticide or when you put some chemicals which is not broken down very easily, what happens? If something is not, for example, you have put waste. Okay, and you know that plastic waste cannot be broken down easily. You need chemicals and things like that, but it is just there. If people come and throw it again and again, what will happen? It will accumulate. What will happen? It will pile up, it will accumulate. And when this happens in the case of biological substances, when this happens, normally in our case, we call this as bioaccumulation, where basically the chemicals which are not going to get broken down really easily, they are going to get accumulated. Okay, they are going to get accumulated. Why they are getting accumulated, my dear children? Because they are not broken down easily, which is why they are getting accumulated. Now, what impact will it have on human beings if they are acquiring the last trophic level? So, for example, if this is plants, my dear children, and plants, for example, are being eaten up by, what do you say, what can I put over here? Suppose it is eaten up by or. The best example I can put, goat, and goat is eaten up by human beings. So what happens is that when we are spraying pesticides on the plants, okay, suppose there are 5 plants. Suppose there are 5 plants. On the 5 plants, I am putting pesticide. Each of them, on them, I have 3 mg of pesticide that I have put. Okay, 3 milligrams of pesticide that is there. Okay. So basically, over here, separately, if I see, each of them has 3 milligram on them, the plants. The goat which is going to eat all the plants, my dear children, the goat which is going to eat all the plants will have 15 mg of this particular thing, of this particular chemical, toxic chemical, because it is getting accumulated over here. The plant is not able to break it down. And the same thing, when the goat eats all the 5 plants, what happened? It has 15 mg of this particular chemical. Now what happens to a human being? Suppose it has 2 goat. Okay, and this goat also has 15 mg. So 15 mg plus 15 mg, 30 mg of thing is being taken up by the human beings. So you see, the goat is also not able to break it down. It is getting accumulated. So you see, when you go above the trophic level, there is something happening, and this is known, this something that you see is known as biomagnification. As in, biomagnification as in when you go above the trophic level, the amount of the toxic substance which is there, it is getting magnified, it is getting increased. This is known as biomagnification. And this is a result of what? Bioaccumulation. And hence, what will be the impact, my dear children? We will be the people who would be most affected, because we are the ones who occupy the highest trophic level. The highest trophic level. So everything bad which is there in their body is accumulated in them, and we are going to take in. So obviously we are going to get accumulated. Okay, we are going to get not accumulated, we are going to get disturbed because of that. So pesticides is a non-biodegradable thing. Okay, it keep getting accumulated at each trophic level, persist for longer time. Obviously, it is not getting broken down, so it will be there. And thus, last trophic level has the highest concentration. Humans will have the highest concentration of pesticides and leads to biomagnification. This is because of biomagnification actually that humans have what? Highest amount of pesticide, which is why they won't be able to live properly. Get it? This is how you need to answer in points, my dear children. Any 2 points is required over here. Write it properly. Okay, write it properly. Pesticide is going to get accumulated at each trophic level as it persists for a longer time, and thus and hence the human beings who occupy the highest trophic level would be having the highest concentration as well, and this is known as biomagnification. Cool. So this is also done. Now section C is time, which is the section for 3 marker questions, my dear children. Okay, again you would be having 2 of them over here. Let's see. Water is used by the leaves of the plants for photosynthesis, but rather than watering the leaves, we water the plant through the soil. How does this water reach the leaves of the plant? So people sometimes, when I was a kid, I used to water plants like this. I used to make them take bath. But my father used to come and say that no, you should not be putting it there, you should be putting in the soil region. I didn't understand then until I reached a class where the teacher explained me really well that how does plants actually take in water. So the vascular tissue which is responsible for taking in water is what, my dear children? The vascular tissue which is there in the plants who is responsible for taking in water, it is xylem. And when I call it a tissue, that means it is made up of different cells. Okay, it will have tracheids. Tracheids are one type of cell, and then it will have vessels, and then other things also. Xylem parenchyma, sclerenchyma, stuff like that. So there are so many cells which makes up this particular tissue that you see over here. Now this xylem, okay, is present from the root tips to the plant top. Is present from the root tips to the plant top. Okay, so suppose I am making a pipe like this over here, or probably I can draw it better by making a plant itself. There's a reason I'm drawing like that. Please give me some time to make you understand that as well. Okay. So the xylem which is there, children, it is going to be there up here. It is going to be there up here as well. Okay. Now the thing is, first thing, there are 2 ways that the plants acquire water. One is the root pressure theory, according to which what happens, plants always need a lot and lots of ions. Okay, they need a lot of ions in them for making of some pigment or things like that. Chlorophyll itself is made up of nitrogen and stuff like that. So what do? They take in the ions from the soil. Okay, what do? They take in the ions from the soil, they take it inside. Now it is inside. So the concentration of ions which is there is way too much. It is way too much. And when this happens, you are basically creating a situation which is known as hypertonic situation. Hypertonic as in when the amount of solute is way too much within something. Okay. Solute as in ions over here is way too much, whereas when you talk about solvent, it is way too less. So ions are being taken up with the help of active transport. They use ATP, they take it in. Even against the concentration gradient, they take it in. Even though if it's more inside, they will still take it in. So what will happen? Since the solute is way too much, it is creating an hypertonic situation, which is why the water will slowly move inside. Okay, what will happen? The water will slowly move inside, and when the water molecules, they move inside, what are they going to do? This water molecule is there, this water molecule is there, this water molecule is there. All of them, they are going to create a push. Okay, they are going to create a push, which is why the water which is there, it is going to get pushed upwards. So root pressure theory is responsible for giving a pushing force in the xylem vessel. Okay, but do you think it will be enough for the plants who have long heights and stuff like that? No, this root pressure theory would not be enough. So at this point of time, what is the next thing that can be done? The next thing that can be done is known as transpirational pull theory that works over here. What happens in transpirational pull is that basically the leaves have stomatal pores. The leaves have stomatal pores, and when sun is bright, that is during the daytime, okay, when the sun is bright, what will happen? These water molecules which are there, they will escape through the stomata in the form of what? Water vapor. In the form of water vapor, and this particular process is known as transpiration. But there is a thing about water molecules that they do not know, and that is water molecules are kind of very much close to each other. They have cohesive force between them. What force, my dear children? cohesive force between them. So suppose one water molecule, it jumped outside. What will happen? The other one is going to pull the other molecule which is down, and now it is going to get pulled upwards. Okay, you also come with me. That's how it works. So there's a cohesive force between the water molecules, which is why when one guy is pulled, the other guy also comes in. And also one more thing, the water molecules and the vessels that they are in. For example, I am a water molecule, I'll be standing like this. So there will be a bonding, a temporary bonding that is there between this and this, such that it will be like I will be moving up like this. So the Hrithik Roshan step in opposite way. That is how it happens. So this is known as adhesive force, my dear children. This is known as what? Adhesive force that is also there. So because of the pull, anyway sun is there, water is there, if stomata is open, it will go out in the form of water vapor and this will create a pull. So transpirational pull theory is going, or basically transpiration is going to create a pull, which is what is meant in transpirational pull theory. So it happens in the daytime. In the night time, what is the one that is helpful? It will be root pressure theory, because sun is not there, obviously transpiration would not be happening. So these are the two things in which way the water is going to reach the leaves. But when you talk about reach the leaves, or basically I can say these are the two ways in which water is transmuted. But when you are asked about how this water reaches the leaves of the plant, then you have to talk about transpiration pull theory. This is the only thing that will help the water reach the leaves, because the root pressure theory would not be enough to make it reach the leaves and all. Get it? So you have to mention about this mainly. And if you asked about how does water transportation in xylem take place, you can explain both. Okay. So let's see the answer, how it is written. In plants, the water is absorbed by the plants from the soil through the roots. Okay, first thing, xylem tissue of the roots, stems and the leaves are interconnected to form a continuous system of water conducting channels. 1 point. Okay, so you told about that the water is taken from the soil, from the roots, with the help of what? With the help of the roots. And you also said that the xylem tissue which is there, it is going to form a connecting link. Okay, they are in continuous system, which helps in the water conduction easily. Now next thing, during the day when the stomata are open, the transpirational pull becomes a major driving force. It is not the only driving force, it is a major driving force for the movement of water in the xylem. Now evaporation of water molecules from the stomata creates a suction which pulls water from the xylem cells of the root to the stem and then to the leaves. So these are the points. Okay, in the second point, they told the transpiration pull is the one. The third point, they explained how it works. Okay, so you get 3 marks for this. Explain in points, underline what is important, for example, transpirational pull and stuff like that, that is through the roots, from the soil, everything. Cool? Chalo. Next question, but before that, let me just turn on the AC. It's pretty hot up here. I don't know why I didn't turn it on before. Yeah. So 30th question. In a family of 4 individuals, the father possessed long ears and the mother possessed short ears, my dear children. Okay, let's see. Let's decode. Father has what? Long ears. And what about the mother? Mother has short ears. Cool. Now what? If the parents had pure dominant and recessive traits respectively, okay, they had pure dominant and pure recessive trait. That means, for example, if long ears is the dominant trait, then it will be denoted as capital L. Then father has capital L and the mother had small l, which is what is written over here. That if the parents had dominant and recessive traits respectively, pure dominant as in when you are talking about this. How did I come to know that the capital L, that this particular long ears is dominant one? Because it is long. This is basically larger size, okay, and also it is mentioned like that itself, usually, okay. Now, and if an individual is married, okay, traits respectively, and the F1 individual, that is the product of this guy, okay, let's see the product. So over here we have the parents. Let's make the gametes. Should I make it here or here? I think I should be making it here. So we have capital and small l, okay. The parents which are there, they would be having capital L in the gametes and small l in the gametes. Now, the progeny that will be formed, that will be having what? Capital L small l, capital L small l, capital L small l, and capital L small l. All of them basically have capital L small l, capital L small l, capital L small l. And L is dominant, so basically all of them have long ears. Now we are asked the second part of the question. And the F1 individual, that is this individual, this is the first generation, okay. The F1 individual is married to an unrelated individual of the same genotype. This is the genotype, my dear children. This is this thing that you see, the genetic makeup is known as genotype. So you said the particular person over here is married to another person who has the same genotype. So basically now you are going to talk about this situation. Let's see the situation. So capital L small l and capital L small l. Okay, this is what the parents now. This is the parents now. What do you have in the gametes? In the gametes you have capital L small l, capital L small l. Okay, and what is a progeny, my dear children? Progeny is capital L, capital L. Okay, capital L small l, capital L small l, and small l small l. So you have capital L, small l, and both of them are different ones. Okay, then calculate the ratio of genetic makeup of F2 generation. This is the F2 generation, and we have to calculate the ratio of genetic makeup. That is, you are talking about the genotype. Genetic makeup is the genotype. Appearance is what? The phenotype. So you see, one of them is pure long ears one, okay, pure dominant one. One of them is pure recessive one. So I'll say one capital L and one small l small l. Now what about the other things? Two of them, they are heterozygous. They are not pure. There is one allele, there is a one more allele. There are two different type of alleles. So I'll say it is like this. This is the genetic makeup, my dear children. This will be the genetic makeup of F2 generation. You have to show the cross. Again I will explain. Okay, in a family of 4 individuals, the father possessed long ears, the father had long ears, and the mother possessed short ears. Okay, this is small l, small l. Now, if the parents had pure dominant and recessive traits, if the parents had pure dominant as in capital, they are homozygous, the alleles will be same. So, father has this alleles and the mother has this alleles. Then the F1 individual, whatever you get, how do you find the F1 individual? You write it in the gametes. Okay, listen, this chromosome can get capital L, this chromosome can have capital L. Any of these chromosomes will be going in, you know, half the number of chromosomes goes to the gamete. So, any of these will be going, which is why we write this and this and this and this. So, suppose the sperm carrying this fuses with the egg carrying this, you get capital L small l. Whatever you do, you get capital L small l. This is the genotype of F1 generation, my dear children. F1 generation. Now, when you are crossing it with this person who has the same genotype, okay, how did you find the same genotype? You found it like this. Okay, now you cross it. Again, what do you get? You get capital L, capital L, capital L small l, capital L small l, and small l small l. What is the genotypic ratio? That is genetic makeup. That is what is being asked, right? Ratio of genetic makeup. So that is genotypic ratio. So it is 1:2:1. If you are asked the phenotypic ratio, what will you say? How do they look? So basically, this also has long ears since both of them are dominant. This also has long ears. This also will be having long ears, and this will be having short ears. So I will say it is 3:1. The phenotypic ratio, it is 3:1. 3 of them have long ears and 1 of them have short ears. But you are asked about genotypic ratio, so you have to just write this. With this, you get 2 marks. Okay. Now, if the father had short ears, ulta, if the father had short ears and the mother had long ears, and both the parents are homozygous for the allelic pairs, same, homozygous as in pure, as in same, they would be having same. What effect it will have on the ratio of the genetic makeup in F2 generation? Same thing will happen. No, I just interchange this thing. Instead of writing it like this, I made it ulta. The father is small l, and the mother is, so same thing I will have, there won't be any change. You get 1 marks there also. Okay, this will be the same ratio that you will be getting. Okay, so how do you write the answer? Any letter which clearly indicate dominant and recessive, example capital L or E for any other trait. You can use anything like that, and now you are explaining. So you fuse them, do not write like this, write how I showed you properly. If you want to know, I'll write. So this is parents, my dear children. You have to write like this and then form the gametes and then form the F1 generation. In the parents, write capital small l, write father or mother who is there, for example father and mother. In the case of gametes, make a circle and show it like this. These are the gametes. In F1 generation, you have to fuse it, make things like this. Okay. Again, capital small l, capital small l, capital small l. This is the F1 generation. Now write, they are all having long ears, they are all having long ears, or if you write genotype is equals to capital small l, phenotype is equals to long ears. Now make the F2 generation similarly, make the parents different, write F2 over here instead of F1, that's all. Rest everything is the same, okay. No change in the ratio of F2 generation will still be this. Okay, cool, chalo. Next, section D, which is a 5-marker question, my dear children. Let's see that. So, attempt either option A or option B. So, this is the option A that we are having, option B I have not shown, I will show you later, okay. So, the question says, the image below, that is this, shows a banana plant which is growing with the help of suckers. What do you mean by suckers? I told you there are extra structures which are there up here, okay. You have suckers like this, okay, which is not the actual part which is responsible for reproduction. So, it is a vegetative part, my dear children. It is a vegetative part. Usually, you know, flowers are the one that is responsible for reproduction to happen properly. Yeah, because flowers will be having anther and stigma and everything like that. Okay, basically the carpel and the stamen, everything like that. But here the flowers are not the one that is going to help with reproduction. Who is going to help? Here it is suckers. So, which is growing with the help of suckers. That means here it is showing asexual mode of reproduction. And what do you call asexual mode of reproduction in the case of plants? You call it vegetative propagation. So, these suckers are small plant stem outgrowths which can be separated from the main plant and planted separately, and they will grow into a new plant subsequently. Give the name and the type of reproduction that is shown in the image below. What do you mean by this type of reproduction? Give the name. What is this? Vegetative propagation, my dear children. What is this? Vegetative propagation. Okay. And type of reproduction is asexual mode of reproduction. Here, where is the vegetative propagation, and it is what? Asexual mode. Now, list 2 advantages of the farmer that the farmer will be having on using the type of reproduction mentioned above. What are the 2 advantages? Basically, this particular farmer would be getting plants in a faster rate, okay. Production will be fast. Even seedless plants can produce, you know, seeds are the ones that is responsible for basically the flowers which are there. It is going to make a seed, and seed is the one that will be giving rise to a whole plant. But here with the help of seedless varieties also, banana also, you know, it does not have a viable seed, right? So, without the help of viable seeds also we can produce things. So, we say even seedless varieties can reproduce properly. That is the thing. There are 2 advantages. You can mention these 2 advantages. Okay. Now, the above plant produces male flowers. This plant that is said, it is producing male flowers. That means it just has the stamen. It just have the stamen, my dear children. Stamen is made up of what? Anther and filament. So, then what's the problem? Let's see what's the problem. Explain how this plant will be involved in the process of pollination. If at all sexual reproduction has to happen. If at all sexual reproduction has to happen, then how will it undergo that? It just has the male part. Where is the female part? It does not have the female part. So, what can it do? What can this particular plant do? How can it do pollination? Can it do self-pollination, my dear children? No. It cannot do self-pollination. It does not have what? The other part, that is carpel. The female part is not there. So, what can it do? Obviously, there is only one choice that it has. It is known as cross-pollination. It is known as cross-pollination. Basically, other plant which has the female flowers, okay, the stamen from here, the anther from here can release pollen grains, and this pollen grains can go to another plant that has the female plant. That is the only way. So, what type of pollination, my dear children? Cross-pollination is going to take place over here. Now, why is the offspring of this banana plant not absolutely identical to its parent plant? Why do you think? It's not absolutely, it's not ekdam same to same of its parent plant. Even though we are doing asexual mode of reproduction, still it is not exactly same. Why so? Why do you think so? So, in asexual mode of reproduction also, basically, you know, the cells which are there, they have their own DNA. create copies of their DNA, right? To give to all the cells which are there. To give to all the cells which are there, you need to make copies. And this copying mechanism of DNA, my dear children, it's not error free. It is not error free. Okay, there will be errors happening. And what do you mean by these errors? These errors means there will be changes in the genetic character. Suppose this is the DNA that you had, you made a copy, but this copy had certain changes here and there. And these changes that you see is going to lead to little amount of variation. Very little amount of variation, but still it will be variation. So why is the offspring of the banana plant not absolutely identical? Because of these things. While copying the DNA for creating more and more cells, each cell should be having DNA, right? So for making a copy, there will be DNA replication that will be taking place. DNA replication or DNA copying mechanism, okay? DNA replication takes place, which is not an error-free method, which is why there will be certain changes in the genetic material which will lead to what? Variations. Okay. Now let's see the answers, how it is given in the marking scheme as well. So over here we have the marking scheme. Vegetative propagation is the one, and asexual reproduction I told you. 0.5 marks for that. So more crops in the same time interval, I told you production will be more. Genetically identical, yes, they would be more. For in sexual reproduction, my dear children, you are basically having two parents. Now two genetic material is coming together, so obviously the offspring will be kind of a mix of both. So it will be different from both the parents. So if I have a good variety of banana, I just want that banana. I don't want to create more different varieties over there. You get it? Because it is already good. So genetically identical plants are also preferred when this plant is already really good. So asexual mode of reproduction, even though it has little amount of variations, it has very little, I told you, does not have variations like sexual mode of reproduction. In sexual mode of reproduction, the parents, basically my parents gave birth to me, I am a mix of both of them. I do not look exactly like any of them, right? I am a totally different organism. Why? Because I have both their DNA, my dear children, which makes me quite unique in my own way. You get it? But if it was asexual mode of reproduction, for example, when amoeba gives rise to more amoebae, everyone looks the same only because they have very little genetic differences that is there. Okay? So more crops in the same interval, genetically identical, flower, fruit faster. Okay, no need to depend on pollinators. Yeah, that is also one more thing. No need to, they do not need to depend on pollinators for reproduction to happen, and also seedless varieties can produce. So instead of writing this, you can write seedless varieties can reproduce. Cool? Now, cross pollination, the pollen from anther will be transferred. I told you what type of pollination will be there. If there is only male flowers, my dear children, then you have to do cross pollination. The pollen from the anther will be transferred to the stigma of another banana plant using agents like wind, water or insect. This is 0.5. So you have to talk about cross pollination in a detailed manner. Okay, talk a bit about cross pollination. That is, pollen from this will be transferred to another plant which is having the stigma. And what is the agents being involved? Wind, water or insects. Okay, explain a bit more. Now there would be minor changes, some variations during the process of copying of DNA, which is why there is different. That is why it is not totally identical. Works. Chalo. So that question is done. Now there's a B part to this question. You can attempt that or this, right? Don't go and attempt both of them. So the image below shows a developing fetus in the mother's womb. The developing fetus is connected to the placenta by means of the umbilical. This is the placenta and this is the umbilical cord. Okay, now the umbilical vein and the artery run inside the umbilical cord. This has umbilical vein and the artery which is going to take blood to and from the fetus. Now name two substances that moves through the blood vessels. Can you name two substances that is going to move through the blood vessels? First thing, gases. First thing, gases, my dear children. There will be so many gases which will be moving down in the blood vessels because the baby would be needing oxygen and baby want to remove what? Carbon dioxide. But when you talk about the next thing, what do you have? You have other things as well. For example, you have gases exchange, and also some waste product that will be moving through the blood vessels. It has to move out. Gases, that is oxygen, will be moving there, carbon dioxide will be moving out, waste also will be moving out. Now if the placenta has less villi, how will it affect the baby's growth? Suppose the placenta which is there, it has less villi. So listen to me very carefully. Placenta is formed both with the help of embryonic tissue, that is the embryo which is there, which is going to form the fetus, okay, that, and also the uterine tissue, that is from the mother. So both mother and the babies, or basically the embryos, helping in the making of placenta. And the placenta is basically made with the help of the villi from the embryo's end. Okay, suppose this villi is not completely interdigitated with the uterine wall. Do you think there will be a connection that will be maintained? No. So suppose you have a thing like this which has villi like this. This and the uterine wall has to get connected, that is when you get the placenta. If this is not, it has less villi, do you think this connection will be strong? No, my dear children. So the placenta has less villi, how will it affect the baby's growth? The baby's growth will be drastically affected, as there is no proper connection being made between the uterine wall and the fetus that is being growing there. Okay. Now name the region where the embryo develops inside the female body. Where do you think the embryo is developing inside the female body, my dear children? Where do you think? It is not in the stomach, okay. It is in the uterus. In the female reproductive system, you might have seen there is a region which is known as uterus. So uterus is the place where the baby's development takes place. Explain how this region is adapted for nourishing the baby. How is this region is adapted? So this region, the uterine wall, it has 3 layers. The outer one is perimetrium, which is there. Then we have myometrium, which is the muscular layer. Okay, why do you have muscles in the uterus? Basically, you know, after the baby is formed, it has to move out. At this point of time, you need to contract and relax, lot of things has to happen, which is why you have a muscular layer. Okay, now outside that, there is a very important layer which you call as endometrium, my dear children. What do you have? You have endometrium, and endometrium is basically a vascular layer. It is what? A vascular layer. It has blood vessels and everything around it. So it is a vascular layer. Cool. And this is what? Endometrium. Basically it has a layer which is always there, and there is a temporary layer which is also being made every now and then, every month. So that this particular endometrium which is there could provide nourishment to the embryo which is coming and joining over here. The embryo will be just a bunch of cells initially. The embryo will be a bunch of cells initially. It will slowly come and get implanted. It will slowly get inside the endometrium and stay there for nourishment initially. And then slowly this particular embryo and the uterine wall together are going to form the placenta later on, which is going to provide nourishment to this, such that this guy that you see turn into something like this. You get it? So basically, how is this region adapted for nourishing the baby? This region has thick endometrium layer which provides nourishment to the baby in the initial time, and it is also going to help in the production of what? Placenta, for basically not production, basically formation of placenta later on, which will be providing nourishment in the later phase of pregnancy. Okay. Now, some of the fetal cells fall off into the amniotic fluid and can be collected by careful procedure. The cells were screened and found to contain XY chromosome. Okay, so basically listen to me. This fetus which is there, it is covered by amniotic sac. It is covered by amniotic sac, and this amniotic sac is filled with a fluid, which is known as amniotic fluid. Okay, and this amniotic fluid will be containing certain cells which are shedded off from the baby's body, okay, the fetus's body. So these cells are there. If I examine the cells, I have nucleus. If I examine the nucleus, I have chromosome. And then it was found that it had XY chromosome, my dear children. What is the sex of the fetus? If it is X and Y chromosome that is there, that means it is not a perfect pair. Why it is not a perfect pair? Because Y chromosome is chutku. X chromosome is a normal size one. Y chromosome is chutku, which is why I call it imperfect pair. And this is found in the case of males, my dear children. This is found in a male child. Whereas in a girl child, it will be X and X. Females have X and X, males have X and Y. Now, how is this prenatal sex determination misused? How do you think this particular process is misused? What do you call this process as? You call this process as amniocentesis. Amniocentesis. How it is used or how it is misused? Actually, it was used since we can find the chromosome and everything. So it was used to find the genetic abnormalities. Okay, suppose a baby which is going to be born has genetic abnormality. Now a choice is given to the parents whether they should be having it. If at all it will be better, if it will be good for the health of the mother as well. A lot of things are being examined with the help of amniocentesis. But people start misusing it as as they could recognize the sex of the child, and since if they were finding that the sex of the child is XX, that is a female child was being born, what they were undergoing? They were undergoing abortion. Okay, they were undergoing abortion, my dear children, and this increased female feticides. What do you call by female feticides? As in killing of the females. The female feticides killing of the female babies, okay? It increased way too much because of amniocentesis. So how is this prenatal sex determination misused? It is misused as people were trying to determine the sex of the child, and then if it was a girl child, they were killing it, which is known as female feticide, and was increasing very drastically, which is why it was kept, the whole procedure has been banned now. Amniocentesis is banned now until and unless there is some strong reasons to do so, okay? In India, I am talking about India. So, yes, name two substances that move through the blood vessels. You have nutrients, glucose, oxygen and waste, any two you can mention. Now, if placenta has less villi, how will it affect the baby's growth? Less surface area for nutrient to pass from the mother to the embryo, slow growth will be there. Okay, there will be slow growth, it is totally not interdigitated. So, which is why it won't be proper. The placenta formation which is there, it won't be proper. Okay, or basically the exchange of gases or nutrients, whatever there, it will also be not properly done. Now, where is the place where the embryo develops, my dear children? It is the uterus, and explain how it is adapted. So, it is the uterus and it has thick lining with rich supply of blood vessels to nourish the embryo, because it has a vascular layer. And what do you call this vascular layer? Vascular layer as in the one that has blood vessels, okay, and this is known as endometrium. Now, what is the sex of the child? It is male. And how is the prenatal sex determination thing misused? As I already told you, misused as if the fetus is the female, some people engaged in aborting the child, leading to female feticide. This is how it is being misused. Works? Cool? Chalo. Section E, the last question of the day, my dear children. Section E, 4-marker question. Let's see, it's a case study based question. Okay, so Mohan and Rohit observed that the shoots of the plant growing in shade bend towards the sunlight, whereas leaves of the touch me not plant fold and droop soon after touching. Droop as in they close, okay, they got dull. So they were curious to know how these movements occur in plants. In order to help them understand the movements in the plants, answer the following questions. Attempt either subpart A or either subpart B here in this particular question, okay, and then rest of the things you have to answer anyways. So these both of them, they carry 2 marks each. You can either attempt this or attempt this. Rest of the things anyway you have to attempt. The one thing, like, you just have to attempt this question and that's all, or this question, these questions. It's not like that. From this subpart you can choose one. Okay, what causes the bending of shoots in the plants as shown in figure A? So what is that something that causes the bending of shoots, my dear children, in the plant A? It is, first thing, this is known as phototropism. Phototropism, where the stimuli is light, and the plant is going to move towards the light over here. It is going to move towards the light. Now, who is responsible for this? This is a 2-marker question, so you have to explain it. You say auxin hormone is the one that is responsible for it. How you can make them understand? You can make this thing over there, okay. So, suppose this is the plant tip. You have this sunlight over here, okay. Auxin hormone is up over here, uniformly distributed throughout the area, okay. It is uniformly distributed throughout the area. Now, what happens? The second scenario will be, this particular auxin hormone will be moving from this side to this side. So, all of them, majority of them are going to be towards one side, my dear children, and the sun is still up. So, auxin hormone basically to the area where light is not reaching, okay, it moves to that particular area. Now, when it reaches to that particular area, what is the thing that happens? When auxin is getting accumulated, okay, it is going to make the cell elongate itself. So, if the cell was like this, it is now going to be like this. There is an elongation that will be taking place. Now, you only think, if it is getting elongated only towards one side, So the cells were normally like this, but now it is not like this. Now they have got elongated, so the structure becomes totally different. And since one side is elongated, other side is still the same chutu cells. So don't you think it is going to bend towards what? The sunlight, my dear children. Yes. So auxin, you don't make like this, okay? I don't know why I made it up there, but yes. So you just make it like this. That's enough for you guys. So it moves towards that side such that the sun which is there, it can provide light towards this side as well. And this happens because of what? Unequal cell elongation. On one side it is getting elongated really well, on the other side it is not, which is why it is happening. In 3 of the cases I have made different color suns. Okay, so sorry, but I hope you understood that. So that is the thing. Okay, now what causes the folding of leaves in Touch Me Not plant as shown in figure B? What causes the folding of plants in Touch Me Not plant? First thing, it is not a tropic movement. What do you mean by tropic movement? Tropic movement means when the basically the direction of the response is in direction of the stimuli. Okay, it is known as tropical movements. For example, the sunflower moving like the sun. Okay, so sun is here, the sunflower will also move according to the sun. So this is the stimuli, this is the response that it is showing by moving. It can move away or it can move towards. Okay, so if it is showing positive, you call it positive tropism. If it is showing negative thing, that is moving away, you call it negative tropism. That is what you mean by tropical movements. But there is other type of movement also which is called as nastic movements, my dear children. Nastic movements have nothing to do with the direction of the stimuli. For example, I touch a Touch Me Not plant. The Touch Me Not plant does not come towards me or away from me. It just closes off. So it has nothing to do with the direction of the stimuli. Okay, so there is no directional thing over there. Okay, it is not done according to the direction of the stimuli. That is what you mean by nastic movement. And Touch Me Not is showing thigmonastic movement. What is it showing? Thigmo as in touch. Okay, thigmonastic movement, my dear children. And why does it close and basically why does it close the leaves, or basically why the folding of leaves happen? Because it has a region, the leaf which is there, no, it has a region like this, a small swollen region like this, which has a lots of water in this. You call this region as pulvinus. So while we touch, there are so many hormones and everything getting activated, the ions and all, so which is why it loses water. And when it loses water, what happens? The leaves which are stiff like this, it will now fall down. So it happens due to loss of water from the pulvinus. And this is a nastic movement. Nastic movement is a non-directional movement. Okay, it is a non-directional movement. Tropic movement, it is directional movement, my dear children. Cool. How do you frame the answer? Let's see that first. So when growing plants detect sunlight, a hormone called auxin synthesized at the shoot tip helps the cells to grow longer. When the light is coming from one side of the plant, auxin diffuses to the shady side of the shoot. Shady side as in the one that does not have light. Okay. This concentration of auxin stimulates the cells of the shoot to grow longer on the side of the shoot which is away from the light. Or you can just directly say it causes unequal cell elongation. Thus plant appears to bend towards the light. Now Touch Me Not plant. Leaves of Touch Me Not plant respond to stimulus by showing growth independent movement. It is not a growth movement. Okay, it is not a growth movement. When I touch a Touch Me Not, just closing. Does it have anything to do with the growth? No, my dear children. But when you talk about the phototropical movements and all, it's a growth movement. The plant is growing towards it. You get it? So it's a growth movement. That one. This is a growth independent movement. And it is not a directional movement as well. These plants that are there, they show electrical chemical means to convey the information from cell to cell. The plants, they actually have this thing, electrical chemical as in basically there will be ion exchange and things like that, the chemicals being released, which is how they perform. Now movement happens at a point different from point of touch. Okay, the movement which is there, it will be happening throughout the plant, but I touched only at the tip, right? So plant cells change shape by changing the amount of water in them, resulting in swelling or shrinking, and therefore in changing the shape. This, you can just directly say the plant cell basically droops because of loss of water in the pulvinus. In what, my dear children? In the pulvinus. That is how they work. It's simple. Okay, and all the plants, it's not just this plant, okay, any plant works in they have electrical chemical means to convey the information, because plants do not have nervous system like us, right? So they just have to depend on ions and hormones and water level and everything like that. This is how you need to explain the answer. Now, over here, next, compare the movement. Okay, compare the movement of growth of the pollen tube towards ovule with the movement shown in part A of the above picture. So this is phototropism. Okay, now when the movement of pollen tube happens, my dear children, I'll tell you what will happen in there. So suppose this is the ovary, and you know there is ovule as well. You know there is ovule as well. Okay, so what is going to happen? The pollen grain, when it's going to come and land up over here, okay, it will now form a pollen tube. It will now form a pollen tube. But how does the pollen tube know that? You have to go inside. So in the ovule, there are so many chemicals which are being released over here. These are known as chemoattractants, okay, chemical attractants, chemoattractants. So they are being released, and on the basis of this, so the pollen grains has a male gamete over here. This male gamete will slowly come through the pollen tube and fuse with the female gamete, which is right up over here in the ovule. So it's a chemical that is being released which causes the formation of pollen tube, and you should be knowing one very important thing. This is known as chemotropism. This is known as chemotropism. Why? Because it is moving towards the chemical and it is showing positive chemotropism. Since it is going towards, here chemicals are the stimuli. In the case of plant growing towards the sunlight, light is the stimuli. Here chemicals are the stimuli, and this is known as chemotropism, and this is an example of positive chemotropism. Okay, when it is moving towards the chemicals. I hope you understood the difference between that and this. Okay. So compare the movement of growth of pollen tube towards ovule with the movement shown in part A of the above figure. So over here, growth of pollen tubes towards the ovule is an example of chemotropism, whereas bending of shoot towards the sunlight is an example of phototropism. Works? Now, compare the movement shown in figure B with the movement of body parts in the animals. So how can you compare this and this? Straight away, let's go to the answer and see. So although both plants and animals show electrical chemical means to convey the information from cell to cell, but unlike nerve cells in animals, there is no specialized tissue in plants for conduction of information. That is there. In our case, we have a nervous tissue, we have a nervous system which have nerve cells like neurons, okay. They are responsible for conduction of information. Can you expect the same from the plant cells, children? No. They have electrical chemical signals that is happening, but they do not have specialized cells for conduction. Okay, they do not have specialized cells for conduction. That is the first difference that is what we have from the plants. Now, in animal cells, change in shape occurs because of the specialized proteins found in muscle cells. Okay, plant cells change shape by changing the amount of water in them. If you are comparing B, in B it is loss of water, which is why the plants, basically the leaves are drooping by, right? So obviously if you compare between us and the plants, okay, why do we change our shape? We change our shape whenever there is change in the shape of the proteins, or probably when there is too much accumulation of fat and stuff like that. Whereas when you talk about plant cells, okay, so if you're talking about animal cells also, the animal cells, okay, if you're talking about cells, it is changing their shape. Why? It is changing, it is going to change their shape. Why? Because it has some proteins being made, or some proteins having some modification in it. But when you talk about plant cells, I hope I spoke about animals, yeah. When we talk about plant cells, in them it is happening because of loss of water or gain of water or things like that. Okay, so even though the second point does not make any sense, it's just there, so we have to anyway just go through it. Okay, but this is the answer that they have put up. Cool. And with this, we are completing all the questions from sample paper 1 that is given in this particular year. I hope it was helpful. If it was helpful, then you have to tell me, ma'am, we got to understand it, and I would be really happy seeing your comments, my dear children. So do not forget to put down your valuable comments and tell it to your friends that we are solving sample papers. Help them also, they might be in need, yeah. And that's how things work. When you help someone, you get help from somewhere. So do good to others, people will do good to you as well. So that's all, my dear children. Do not forget to like, share and subscribe, and also subscribe our cute little channel, that is Diksha Vedantu. See you with the next session, which is going to be there on Wednesday for science. For math, there will be sample paper tomorrow, so be there. Okay, bye bye, take care.